want you to do is we're going to be working in groups of 3 for this whole session. So I want you to get yourselves clustered around in groups of 3, and we're going to work on these worksheets. So today this is a class in virology, so this is an upper level, mostly juniors and seniors, occasionally a few graduate students will come in here. And right now we're in a theme, we're talking about Ebola, and then we want to get into the idea of tracking. So how do we know where did the outbreak come from? And so we're going to do an activity where we're going to be working with some genetic sequences, and so they're going to be working in groups trying to line up sequences and see how we match things up and try and make connections between DNA sequences to understand the process by which we can use genetics to trace epidemics. So let's hear how you group things together. So
we saw that they tended to only have a C mutation and a T mutation.
Created one by itself.
One that just couldn't fit in with anything.
I guess we grouped it a little bit differently. We just had the first C and T mutations.
You could kind of sequentially see where the virus traveled to, like if the group 2 ones were, west of the group 1 ones, and try to predict where it might spread.
This information and then take that to the treatment level. All right, thoughts, questions, comments? When I usually get end of course feedbacks, usually the things that are most pointed out are doing some of the active learning things, but the vast majority of students are very much in favor of doing something where they're working in groups and not sitting in a lecture, so they're actually actively doing something, either trying to figure out a problem, trying to solve a problem, trying to understand the basic data that's involved. So I think it's been a very positive response to these.